Hello, I'm Neil McDonald, head of the Signaling and Structural Biology Lab at the Crick Institute.
I'm Attila Csikasz-Nagy, a senior lecturer in computational and systems biology at King's College London.
And I'm Nate Goehring, head of the Polarity and Patterning Networks Lab at the Crick Institute. Thanks for giving us a chance to introduce you to our work and the focus of the two joint PhD projects for which we're recruiting this year.
So, how do cells know up from down, or their front from their back if they're moving? And how do they self-organize into regular patterns to partition the organism into discrete compartments? Well, at least part of the answer is they form polarized cells with distinct regions of the plasma membrane having a unique identity and unique properties. A variety of examples of polarized cells are shown in this slide. And what's remarkable is that the same proteins are being stained in each case, revealing that the same conserved components are involved in establishing polarity in each context. An example of the consequence of distinct polarized membranes is shown for stem cells that give rise to two daughter cells when they undergo asymmetric cell division, each daughter cell with a different cellular identity, shown here a neuron and another stem cell. So what happens when cells lose polarity? Well, for epithelial cells, this can lead to cancer, and indeed a disrupted organization of epithelial cells is a hallmark of metastatic cancer.
One of the core pathways that drives cell polarity in animals is the so-called PAR network. And as you saw in the previous slide, a key feature of PAR polarity is the localization of polarity proteins into membrane domains that serve as a compass to guide downstream processes. In the case of the C. elegans zygote shown here, which is our model system of choice, two groups of PAR proteins mark domains that divide the anterior and the posterior of the embryo. And it's these domains that drive the asymmetric segregation of fate determinants that will differentiate the daughter cells when the zygote divides. And this ensures one follows a somatic program while the other can retain germline potential. Now, this is thought to work through cross-inhibition between these two groups, each inhibiting the other's localization of the membrane. However, we're only beginning to understand the complex network of molecular interactions and feedback circuits that drive their self-organization into domains. Some of the key questions we seek to address through these projects include: how do dynamic molecular interactions drive the emergence of cell scale patterns? What are the relevant functional complexes and what do they do? How's the localization and activity of such complexes regulated in time and space, for example, throughout development? And what are the core network feedback circuits and how are they encoded? Addressing these questions require that we bridge fields and link relevant scales of biological organization from the molecular to the cellular. Each of our labs brings a unique set of tools and perspectives. The McDonald lab brings expertise in the structural biology and biochemistry of cell signaling pathways. This includes X-ray crystallography, cryo-EM, drug design, enzymology, and in vitro cell-based assays. The Goehring lab provides expertise in quantitative cell biology and C. elegans genetics. And this includes live cell imaging, single molecule tracking, CRISPR-based genome engineering, all of which we use to explore PAR protein behavior and function in vivo. And the Csikasz-Nagy lab brings expertise in computational modeling of biological systems, especially focusing on spatial temporal behavior of molecular regulatory networks. And between our three labs, we're proposing two projects, each of which will focus on a collaboration between two of the three labs.
The first project will be based in the McDonald and Goehring labs and hosted at the Crick Institute, and will focus on the core PAR polarity kinase, atypical PKC or APKC. We'll address what the polarity complexes look like, where and when they are active in the cell. Our results to date support a model in which APKC activity is regulated through its dynamic interactions with other molecules within the PAR network and at the plasma membrane. Yet the proper regulation of APKC activity in time and space remains poorly understood. We aim to take advantage of our combined expertise in structural, biochemical, and cell biological techniques to identify and characterize potential states of APKC containing complexes. A key aspect of this project will be to design structure-informed reagents to visualize and manipulate these activity states in vivo. to reveal their roles in polarization of cells. Tools including chemical modulators and reporters of APKC activity are available.
The second project will be based in the Goehring and Csikasz-Nagy labs, so between the Crick and King's, taking advantage of the expertise of colleagues and resources at both institutes. We will combine genetic analysis, quantitative imaging, and mathematical modeling to understand more globally how feedback circuits within the PAR network drive patterning of polarity proteins across the cell. The project will build on modeling efforts in our labs and leverage the advantages of the C. elegans system, including considerable prior knowledge about the PAR network, exquisite control of protein dosage and function, and amenability to quantitative imaging. Through links to ongoing work in the McDonald and Goehring labs, we ultimately aim to trace core features of network behavior back to molecular interactions, ultimately aiming to make novel predictions about the biological system that can be tested in vivo.
Our belief is that work at each scale of biological organization will be synergistic, with insight from each helping to inform and guide work at other scales. We envision that the students recruited to these projects will hopefully benefit from working together. They can take advantage of the varied strengths of the individual labs and hopefully complement one another, both in terms of their technical expertise and their scientific perspectives. Because of the collaborative and multidisciplinary nature of the projects, successful candidates will need to be highly motivated, open-minded, willing to work across fields, for example, combining biochemistry with C. elegans genetics or cell biology with modeling, and certainly be confident working together as part of a team. If this sounds like a good fit, I'd encourage you to check out our websites where you can find out more about our science. Thanks for tuning in, and please don't hesitate to get in contact with us by email if you'd like more information.